Thank you all for coming to our seminar this Friday. Today, Dr. Jessica Pruett is joining us. She's at the University of Southern Mississippi. She's currently a research scientist connected with Embrace, the Mississippi Restore Center for Excellence. She started at UCLA with her undergrad and then did her PhD at Georgia Tech with Mark Weisberg's group in chemical ecology before moving on to a postdoc with the University of Mississippi, and then now attached to the Center for Excellence in Mississippi. And she's going to talk to us about some of her work on oyster restoration. So if you could please help me welcome Jessica.
Thank you for the introduction. Can you all hear me all right? Or not known for being loud, but so let me know if I start mumbling. But yeah, so thank you, Jessica, again for the introduction. Thank you all for the invitation to come out and speak with you today. And yeah, to discuss some of the work I've done looking at the influence of abiotic and biotic factors on early oyster life stages. So I'm going to discuss some of the work I did during my PhD and during the postdoc at Ole Miss and some of the current stuff I'm doing with Embrace. So, I mean, for y'all, the oyster really needs no introduction, especially throughout the Gulf, because most of us can appreciate it's delicious in a variety of forms. But more importantly, they form these large biogenic reefs that are really important habitats that provide a lot of important ecological benefits and ecosystem services that we're increasingly realizing are really difficult to replicate, as well as it's really expensive. And so oyster reefs actually represent one of the most degraded habitats worldwide, as for coastal habitats. And so y'all are probably familiar with this study by Beck et al. In 2011, where they estimated that about 85% of oyster reefs have been lost globally, largely due to overharvesting for consumption because they're so delicious, but also because of habitat degradation and associated diseases that come with that. And so as a response, this has really prompted a worldwide effort to use oyster restoration as a practice to help recover those oyster populations and the valuable ecosystem services that they provide. And so this is a map of distribution of oyster restoration projects worldwide, coded by different species. And so in particular, Crassatrea virginica, or the eastern oyster that's found here on the eastern shores of the United States, as well as the Gulf, has really been the predominant number of those projects within this graph. So on the y-axis, we have the cumulative number of research restoration projects. And so these green dots are eastern oyster projects that really begin to sort of exponentially spike starting in the late 1990s. And of those almost 2,000 projects that we found for our database, about 686 of those have been in the Gulf of Mexico since 2000. And this is really as a result of this significant amount of funding that's been made available for restoration projects from the settlement penalties following the Deepwater Horizon Oil Spill that impacted the entire Gulf from the eastern Texas all the way to the Florida Panhandle in 2010. And as of the end of 2020, over $200 million have been allocated to oyster restoration and related projects. Yet, despite these really intensive restoration efforts, there's still been a decline in oyster landings from wild harvest in the Gulf. So this is a graph of the number of pounds of oyster meat that's been harvested since 2000. And you can see it's just been steadily declining to present day, with several states having fishing moratoriums. In particular, Mississippi didn't have public harvest for 5 years between 2019 and 2023. And this is really the result of several mass mortality events that have occurred since Hurricane Katrina in 2005. So this graph shows data from the Mississippi Department of Resources, their dredging data from their fisheries independent stock assessments. And so these solid lines, this is a legal size oyster, so everything above 75 millimeters, where these dashed lines are everything below that. And so we see that populations were able to increase post-Hurricane Katrina. They've been dropped dramatically following the BP oil spill, as well as the 2011 Bonnie Carey Spillway opening. And there was some increase in spat and seed oysters, but those then dropped and declined again following another 2019 Bonnie Carey Spillway opening. So y'all are probably familiar because in 2019, those effects were felt all the way over to Mobile. But basically, the Bonnie Carey is a diversion system that diverts fresh water from the Mississippi River to protect New Orleans from flooding. And so all that fresh water flows into Lake Pontchartrain and it makes its way into the Mississippi Sound and can dramatically decrease the salinity there and have really catastrophic effects on both the oyster populations as well as other estuarine critters that, like it to be a little bit more mixed. And so really for these restoration efforts, especially in systems where the adult densities have severely declined, are really dependent on the success of sustained recruitment of these early oyster life cycles. So oysters, y'all are probably familiar, have sort of these mass spawning events where they release their gametes into the water column, they become fertilized eggs, and then form into their larval stage, in which they'll float in the water column for about 2 to 3 weeks, which point they become these pedovelger larvae, where they'll then seek a hard material to settle on and metamorphose into that sessile oyster stage, in which it takes about 2 to 3 years until they can recruit into the adult population and begin then breeding. And so, however, there's right, the success is really affected by a suite of different abiotic environmental conditions, as well as biotic interactions that can influence the growth and survival of these early oyster life stages. And these are generally less well understood than for adults. And additionally, these earlier life stages can be a lot more susceptible to environmental stressors. And so that kind of segues into the outline for the rest of my talk, which I'll present on the work we're looking at, how do these early oyster life stages respond to water quality stressors associated with freshwater flooding events like the Bonnie Carey Spillway. And then moving on to thinking about some of these biotic interactions and can predation risk cues influence the oyster recruitment process. And then thinking about some of the work I'm currently doing now, where how do we begin to, translate these types of research findings to be better applied to restoration and management decision-making. And so thinking about sort of freshwater flooding events, these can really have cause multiple stressors in estuary environments. So during periods of heavy rainfall, or when you divert large amounts of fresh water from one of the largest watersheds in the world, you're getting these elevated freshwater inputs that can reduce salinity levels suddenly and for extended period of times in estuaries. And so that drops your salinity, which can be stressful, but it's also contributing a lot of nutrients, which can lead to algal blooms and then the associated issues of eutrophication when those blooms begin to die and decay, causing things like hypoxia and acidification. And additionally, river discharges are usually more acidic than oceanic waters. And so that's further contributing to these issues of coastal acidification. And so you have these multiple co-occurring stressors that could possibly have complex interactive effects on these species that are inhabiting often already degraded systems. And so for the Mississippi Sound, freshwater inputs really heavily influence this system. So in this graph on the top here, this shows basically these lines represent 15 different times that the Bonnie Carey Spillway has been open since its construction in the 1930s. And of those times, there's been reported six of those times there's associated mass mortality of the oysters following those events. And so you can see they're corresponding when there's the largest amount of cumulative freshwater discharge, but also these events or when the spillway is open, extending into the May month and this warmer spring waters. So causing, oysters' ability to combat sort of low salinities decreases as temperature rise. Additionally, the amount of localized rainfall can really impact salinity conditions and is really can be really variable throughout the years. And as well as, so the Pearl and Pascagoula rivers contribute a lot of fresh water into these systems as well. And that really varies from year to year, but sort of overall since the 1930s, there's been a slight trend of increasing amount of fresh water in these systems. And so I'm going to talk to you about a study we did looking at, the effects of these flood-associated stressors on larval and juvenile eastern oysters, to kind of tease apart, are there combined impacts of acidification, hypoxia, or low salinity events, to really sort of help us sort of understand, which one of these stressors might be the most problematic, or again, there's always those issues of thinking about synergistic interactions when you start to combine these different stressors. And so we performed these in the lab up in Oxford, Mississippi, basically looking at these 4 different sort of stages of the early oyster life cycle and exposing them to a combination of either low dissolved oxygen, low pH, or salinity in isolation, or in their different 2-way and 3-way combinations, in looking at their survival and growth responses to those. And these concentrations were based on measurements taken during the 2019 opening, as well as I'll show later, they coincide with the conditions we often seen happening in the Mississippi Sound. And so for the results, we're going to have the next few slides are going to look like this, where we have graphs. Where on the y-axis, we'll have some sort of oyster response. So we'll begin with looking at the response of the D-stage oysters that were exposed. So these are 2-day-old oyster larvae that were exposed for 96 hours to these different combinations. And we see that there was so survival was similar across all the treatments, but we did see reduced growth in the hypoxia treatment and the low salinity, and sort of having an additive effect of, when you put those together, of actually negative growth or just barely growth to near 0. And so these pictures show what sort of oysters the larvae looked like in the control treatments, and you can see in the combination of all three stressors, they look a lot less happy and smaller. Whereas, so the next experiment we did was using petabelgers. So these are oyster larvae that were ready to set, and expose them to the different treatments for 48 hours and see how many actually settled on these tiles that were provided, and successfully were metamorphosed to the next stage. And so for these, we actually saw that there was an effect of all 3 stressors, and they sort of actually worked in a slightly antagonistic fashion, but overall they all lowered settlement when exposed. And then for the following experiments, so we exposed, these were day-old oyster spat. So these oysters had just settled onto and metamorphosed into that sessile adult or oyster stage, and we exposed them to these conditions for 7 days. And we actually saw we saw large impacts on their survival. So these graphs, so in this case, we have the proportion. So blue are the proportion that were alive, where red is dead. And the lighter ones are the treatments that were exposed to low salinity. So we see a lot higher mortality when these day-old spat were exposed to low salinities. And additionally, looking at their final shell area, we saw strong decreases in growth following exposure to these low salinity treatments. And there was a slight effect of hypoxia, but not as dramatic as that impact of being exposed to these lower salinities. And just to state earlier, the control salinities were 15, and these low salinities were 6. So, and then for the final exposures, we exposed these were 4-week-old juveniles that had been reared here at Auburn Shellfish Lab prior to exposure in the lab for 24 days. And we see, again, impacts on survival, but not as dramatic as for that initial 1-day-old oysters. But again, strong impacts of the low salinity and hypoxia treatments. And saw decreased shell growth rates when exposed to all stressors. Really, it was still sort of additive, but the strong, actually negative growth when exposed to all three stressors and combinations. And so these pictures at the top show, you can see these control oysters, you can see this growth at the margins here, and when they were happy, but and then when they're exposed to all three of the stressors, you can see they're a lot smaller, sort of that sort of degraded at those shell margins. And so in summary, low salinity had negative effect on all the early life stages tested, but there were slight differences in response between the different stages. So the larval stage, there was no lethal effects, but there was reduced growth and settlement when exposed to lower salinities, whereas at the juvenile stage, there was both, again, decreased growth, but especially on those day-old spat, really strong impacts on really strong negative impacts on survival. And additionally, hypoxia was more stressful for the larvae, whereas acidification strongly decreased the shell growth in those older juveniles. Yet overall, there was no synergistic effects. So the impacts that we're seeing in just the isolated different stressors, we're able to predict the responses at when exposed to all three stressors. And so if we put these results sort of in context of, again, the eastern oyster life cycle and thinking about the implications this may have for restoration efforts in field populations. So even if you deploy culture in area, so if you deploy culture, have reefs in areas with suboptimal water quality, even though you may have adults spawning in areas where the conditions are good, when they sort of larvae move into these waters with these lower water qualities, you're going to see reduced growth in your larval stages, which are going to cause a longer time in the water column, and it'll take them longer to develop, which increases their risk of predation as well as depletes their energy stocks and reduces the amount of sort of competent larvae that you'll have available to actually settle onto hard materials in the environment, reducing the amount of oysters you're getting recruited to either your reef or your culch. And then once they do settle, you see really strong impacts of decreased survival on those initially set oysters, reducing further the amount of potential recruitment you're getting in these areas, and seeing strong effects on reduced growth, which is going to delay the time that you'll get these oysters to sexual maturity, which is overall reducing your resilience in the reefs. And so in Mississippi, as well as across the Gulf, these flood-associated stressors are really a real threat to restoration efforts. So for example, this is in 2021, so there wasn't any Bonnie Carey spillway openings, but there was record rainfall across all three coastal counties in Mississippi, leading to, so this table shows the average salinity during peak spawning months between May and September. And so these are either, 4 of these sites are USGS water quality monitoring stations, or that purple star, we actually deployed out sensors with the help of a local high school down in Bay St. Louis. But you can see through those sites, the average salinity was below 6, which was what we exposed the oysters to in our lab experiments for the entire spawning season. And additionally, all the sites were subjected to sustained periods of low salinity. And so, yeah, it's just, and additionally, and additionally, at that site, we partnered with the local high school. We were able to get measurements on pH and dissolved oxygen. So these graphs show, so these dashed lines were the conditions we exposed in our stressor treatments. So you can see that pH experienced near Bay St. Louis in the Mississippi Sound were within the range that we exposed oysters to, and often dipping below pH levels that were found to be stressful. And additionally, we did see a 3-day sort of hypoxic event towards the end of our sampling period, sort of demonstrating that, these conditions can co-occur and often do in the Mississippi Sound. All right, so transitioning sort of gears into thinking about what are potential biotic interactions that might affect these early oyster life stages. So this question of could predation risk cues influence oyster recruitment? And so as I mentioned before, there's a suite of different parameters that can determine how many larval invertebrate larvae you get that settle and actually recruit into the next sort of population. And and a strong important factor in that is predation. And often for a lot of marine invertebrates, about like 80 to 90% of your larval supply is taken out by larval predators. And so as we're familiar with a lot of the work what the SME lab does, so obviously predators decrease the amount of individuals you have in a population, but just their presence can cause changes in prey behavior and morphology. And so this experiment was thinking about, so we know there's a lot of positive cues that can influence oyster settlement. So adult oysters and biofilms can induce settlement behaviors in oyster larvae. But thinking about, are there negative cues that particularly put off by predators that might sort of deter settlement in oyster populations? And so, because there's a strong influence of to be able to avoid predators, since it has a big determination on your ultimate success. So most suboptimal is death. But basically, this experiment was thinking about if are oyster larvae able to respond to negative predator cues, and if so, what types of cues are used by larvae to avoid predation. And so in particularly, this was work I did during my PhD. So it's been about 6 or 7 years now. That's why I'm a little tongue-tied in explaining it. But so this was work I did on Savannah, Georgia. And so in intertidal oyster reefs, but an important predator present here are mud crabs. And so looking at, during do mud crab cues deter oyster settlement, and are they forward? So during a predation event, you get cues from them, as well as they're eating other oysters, this crush conspecific cue being put off. And so basically, I first did field experiments where I put out settlement tiles in the marsh or within the intertidal oyster reefs. So there's my little settlement tile there, and then surrounding them were these 4 different treatments of either just a control of crushed shell or just the sort of conspecific cues of crushed oysters, as well as mud crabs that were being fed crushed oysters, and whole oysters as just sort of a positive control. And so I deployed these different treatments, set them out in blocks along sort of the mud flats here. And I left them out for 6 weeks, and weekly I'd go out and refresh the cues, as well as take photos of the tiles to be able to monitor the recruitment that was taking place weekly. And so for the results, at the end of the experiment, this is the number of oysters that recruited to the tiles. And as you can see, the number of oysters that have recruited to the tiles were significantly lower when they were near mud crabs being fed crushed oysters, and they were highest near just whole oysters. And sort of looking at sort of recruitment throughout the experiment, there was 2 pulses in oyster recruitment throughout the experiment. And so this is looking at each week, how many new oysters were settled on the tiles in the different treatments. And so during this first spike, we saw that, again, reduced recruitment in the mud crab and crushed oyster, as well as the control. And then in this last one, reduced recruitment next to the mud crabs eating on crushed oysters, as well as the crushed oysters. And then also because I had photos of these, I was able to measure their shell area at the end of the experiment. And based on when they showed up on the tile, could sort of calculate this growth rate. And even though there's not a strong effect on recruitment, interestingly, oyster growth was highest when exposed to just the crushed oyster. And so I then went into the lab to sort of tease apart, how are they responding to these differences in chemical cues from either the crushed oyster or the mud crab. And so I borrowed this experimental design from a paper by Morello and Yun, who looked at the response of mussel larvae exposed to negative cues. And so basically these are each individual choice chambers. So I could put a little competent Petavelder larvae in the center there and introduce one of these different cue treatments in one of the sides, and then just was in the dark and hung out and watched oysters make decisions potentially. And so, yeah, so I either had just the no cue control, the crushed oyster treatment, or I used a hermit crab as just sort of a neutral control. Are they just responding to general crab cues potentially? And then our mud crab cue, and then a whole oyster. And so what I found was that, so we see when they had sort of just no cue on either side, they just tended to hang out in the middle, and they did sort of swim to a different side. There was really no preference. However, when you introduced a crushed oyster, they moved away from that side if they did decide to move from the middle. And then whereas for the hermit crab, they didn't really have a preference for either. And then again, sort of avoiding that side away from the cue for the mud crabs, and we're actually, attracted to that side that had the whole oyster. So in summary, we did see impacts on oyster recruitments which was significantly lower in the presence of actively foraging mud crab predators. However, chemical cues from crushed conspecifics alone didn't affect recruitment under natural flow conditions, and maybe as a result of, either sort of qualitative or quantitative difference in the cue that's being produced. However, there was an interesting impact of seeing increased growth rates when exposed to those injured preys alone. And that could be a potential sort of less costly. So once they're settled there, because in those oysters that were bigger in those treatments, you could it was like a thin shell layer that was being sort of secreted at the margin then. So maybe a potential sort of less costly response once they're already there than just full on avoiding settling in the area. But in the lab, we did see that oyster larvae avoided chemical cues from either the mud crab or crushed conspecific when presented separately. All right, and so we're moving on to sort of this final question and some of the work that I'm doing presently at the University of Mississippi with Embrace. How do we translate research findings to better be applied to restoration and management decision making? So many of y'all are probably familiar with the Centers of Excellence since the Alabama Center of Excellence is hosted here at Dauphin Island. But in case you're not, so these are the Centers of Excellences. So this is from the Restore Act, so portions of the Deepwater Horizon settlement money going towards establishing different Centers of Excellence in each Gulf states to help improve that baseline understanding of ecosystems in the Northern Gulf, which we realized after the spill we didn't really know much about, and really supporting the research needs of coastal communities in each of those states. And so for Embrace is the Mississippi-based Restore Act Center of Excellence. So it's a consortium of the 4 major research universities in the state, with USM being the lead institute and where we're based out of, but also we have researchers at Ole Miss, Jackson State University, and Mississippi State. And so the mission of Embrace has been to really facilitate the sustainable use of Mississippi and the Gulf's important resources through sort of a scientific and comprehensive understanding of different chronic and acute stressors that affect ecosystems within both the Mississippi Sound and Gulf wide. And so given the historical importance of oysters in the state and the severe recent declines, really oysters have been a major focus research priority for Embrace. In particular, how do we support both healthy and sustainable oyster production for both economic, ecological, and social benefits of the citizens of Mississippi. And so to support those efforts, we're using a structured decision-making approach, basically sort of depicted in these 6 steps, but really to help sort of assemble both information that's already been gathered within the Gulf and that we're producing through Embrace Research to help assemble that in an organized manner so it can be a shared resource for the Mississippi oyster community practice. And again, better apply research findings in that appropriate context for effective decision making. So I'm just going to focus on this first step for the rest of our time, which is really kind of where we're at in this process of beginning to really clarify the decision context. And so to do that, we've been using this DPSR framework, or driver's pressure state impacts response. And so this is a well-defined approach that's been used by a lot of organizations, both European and in the United States, that's useful for organizing and communicating complex environmental issues by helping describe the linkages of the social and economic drivers that are causing pressures that create changes in our ecosystems and impacts they have on the services and goods that we like to have and need from our ecosystems, and what are different things we can do as responses to better either promote those or decrease the impact of the pressures into these systems. And so in generating this framework for oyster reef sustainability, it's easiest to start with this oyster life cycle that I've been talking about so far, and thinking about, again, as I just mentioned before, the different abiotic conditions that can impact that cycle with the survival, growth, and reproductive capability of oysters. And those are also influenced, again, by the different biotic interactions. But, these are really controlled by different physical processes that are happening within the water column that can change the abiotic and biotic processes that shape the oyster habitat characteristics that produce these different ecosystem services that we know and love from oysters that contributing to our well-being as humans. And so within this framework, we can think about what are different pressures that humans create to cause change on this oyster ecosystem. Things like, again, our harvesting, removing oysters from the system, dredging, removing habitat for the oysters, but as well as the discharge, the different waterborne or airborne discharges that can take place. And again, the impacts of land use changes. And these are driven for different reasons, by different needs we need as society to exist. And you can begin within this framework to visualize different responses that can be taken by management or individual citizens on these different aspects of the DPSR framework. So for drivers, we can issue different policies or regulations for reduce pressures, create different management applications, or just educating folks in general about the impacts of what they do has on ecosystems. And then within the system level, we can restore or monitor how the system is changing, and then really thinking about what are the different economic values we're getting from these different services. And so with this, generalized conceptual framework, we can begin to hopefully summarize information about specific challenges. And so for in our case, to the Mississippi oyster population. And so as I mentioned in introduction, the impact of the Bonnie Carey spillway is a really important issue for Mississippi oysters. And so thinking about within this framework, so the spillway is a result of benefiting the health and safety of New Orleans, but through that, and then causing hydrological changes to the Mississippi River, discharging that fresh water into the Mississippi Sound. But with that fresh water, we get all of the stuff from what 75% of the United States coming into our Gulf waters. And additionally, with climate change, as a result of the rises in CO2 gases, there's the possibility that we're going to get more frequent openings as we continue on due to increased instances of heavy rainfall events, further contributing to the need for spillways. And so, as I mentioned before and demonstrated with the lab experiments, these different stressors associated with those freshwaters have negative impacts on both the early oyster life stages, but as we saw in 2019, the field populations as well. So basically, 100% mortality was reported during this 2019 spillway opening from monitoring that was being done by Embrace at the time. And through DMR, they reported about 98% mortality on reefs during this event. And additionally, so yes, if you don't have adults, probably don't have any spawning. And additionally, Chet Rakoczycki had settlement tiles out during this time, and I think he had 150 settlement plates out. It only found 2 spat settled onto those oysters. So it was a rough year. And as a result of following the 2019 spillway opening for 5 years, there was no public harvest in Mississippi. And so really post that event, a lot of folks are saying, well, is it even worth investing in restoration in Mississippi if they're just going to open up the spillway again? Luckily, we've now sort of, which it seems is kind of the case looking historically back at the oyster populations in Mississippi. You'll have these spillway opening that kills everything off, but then in these periods of heavy rainfall and really wet years, followed by drought conditions, and then with that is another suite of issues of predators. But then in that, everyone's like the salinities are too high, and we forget about the freshwater impacts. But at least since 2023, we've had really good settlement years, and the fishery's been able to be opened again. But in thinking about, how do we possibly better restore oyster populations that are resilient to those openings? One way is to think about, which is no easy task at all, but how they manage the spillway. So the spillway is managed by the Army Corps of Engineers. And so potentially you can either delay, change the timing of when those openings occur, how long they occur, how many, so the flow rates, it's basically, it's a pretty simple system. They just pull, they have a wall that's blocking it, and then they pull that and the water flows, but you can sort of determine how many different bays you open at a time. And there is an alternate diversion that could go to the west through the Morganza spillway, but then that opens up a lot of issues for all Louisiana oyster populations to the west of the river. But there was sort of a 5-year comprehensive lower Mississippi River watershed project that the Corps was conducting, and we're actually having sort of quarterly meetings that the public can attend, and they're actually looking into thinking about, are there ways they could change both even at the so the old spillway or the old river control, how much they divert to the Chifalaya. So they're beginning to think about those ways, and they're at least now we're talking to folks in the state of Mississippi. There was good sort of dialogue going on. Unfortunately, the funding for that has been pulled, but currently the Mississippi Sound Coalition is trying to push for, and it's got support from the coastal counties in Mississippi, but to get Congress to find funding to reinstate that project. But so hopefully that can, because it was, whether they'll change anything, it was at least nice that they were listening. So that's at least a good step in one direction. But along with spillway management, we can also, and actually the state has been diverting more efforts and interest into restoring the reefs in the eastern Mississippi Sound. So these can Unfortunately, during the 2019 spill, it was kind of a blanket negative effect across, but at least potentially in less extreme years, these eastern Mississippi Sound reefs can serve as potential source reefs for those events. And another technique that the state has done is sort of taking the adult oysters from the reefs before the spillways opening and moving those to the eastern reefs. A good majority of the oysters die during transit, but I mean, still something. And really a big push that throughout the Gulf they've been investing in, and in the state of Mississippi is really encouraging aquaculture. So sort of be able to reduce the amount of pressure on wild caught oysters. And so yeah, so with hopefully sort of that example demonstrates the potential utility of having this DIPSR framework to really be able to communicate with stakeholders, sort of how different results can inform different management responses. And so as we continue to move forward with utilizing the DIPSR within Embrace, begin to continue on this sort of structured decision-making approach, and really beginning to think about different objectives and measures the state we can be focused on in our research efforts to benefit the state. But the state has sort of through this 2016 Oyster Council report and a 2020 recovery plan have really outlined a lot of the objectives they have for the state, which are really the goals of being able to increase oyster harvest, create new jobs and business opportunities, but also not just the economic benefits of the oysters, but really hopefully have a thriving oyster population that can improve the environment. And sort of the approaches to that are improving water quality, creating and restoring reef, and promoting aquaculture. And so for our next steps, begin to think, okay, sort of reevaluate sort of those different alternatives and options that's already being considered and implemented in the state, and are there possible, and thinking about the costs and consequences of those alternatives, are there things we're overlooking or things we should continue to invest in to help benefit the restoration and management efforts. And then within that, what are really the key knowledge gaps and uncertainties that we're missing to help really better inform this decision making. So with that, just think there's a lot of people that have helped and been part of the work that I've shown today, and different funding sources. So for my PhD, and that project was funded by NSF, as well as a GAN fellowship I received. And then so this work done through Embrace and supported by DEQ and DMR. And with that, thank y'all so much for listening.